Good afternoon, everybody. Thank you for attending my workshop called Application of EEG ERPs in Linguistic Experiments. My name is Srdan Popov. I'm an assistant professor in Neurolinguistics at the University of Groningen in the Netherlands, where I also run our EEG lab. So today I will be talking mostly about the application of event-related potentials in linguistic experiments, and I will explain in a second what exactly it is. So I think we can start. So the talk is going to be organized in the following way. I will tell you a tiny bit of electroencephalography in general, a very, very tiny bit. Mind you, I am a linguist, I'm not a physicist or an engineer. I've picked up a couple of things during my PhD and also my research career, but I will try not to talk a lot about things that I actually don't know much about. And then I'm going to explain to you what event-related potentials are and how they're related to EEG, even though I trusted that most of you are familiar with this already. So I'll try not to take too much time talking about event-related potentials in general. After that, I will introduce two ERP components, namely the P600 and the F400, because they are going to be the basis for the talk today. And then we are coming to the actual core of this talk, which is the experimental design in ERP research. I will be telling you about methodological choices that you have to make in ERP research and which choices I consider to be best or most suited for ERP research, or I will give you some references as to where you can look up certain things. And after that, we have a wrap up. So in case you're not entirely sure what today's talk is about, because, well, to be honest, the title is somewhat general, I've decided to talk about things that I'm most familiar with and that I also consider extremely, extremely important. So the goal for today's talk is to draw your attention to methodological choices as well as some technical aspects very, very minutely. I'm not going to talk a lot about that in setting up your own EEG research. Right. So I believe you're all familiar with this. You know what electroencephalography is. You know probably what EEG is. If you haven't done research with it, you've probably seen it in a medical setting, in a hospital or somewhere. But as a general introduction to today's talk, I'm just going to tell you a couple of things. So what EEG does is essentially it records changes in voltage or electrical brain activities at or along the scalp. The important bit is that this is happening on the scalp. We are not doing intracranial recording. That's a completely different thing. We place electrodes on the scalp, and in order to do that, we need some equipment. The equipment that we use, well, the equipment that we used before, you see in the left hand side picture. So that is an EEG amplifier, the RF amplifier that we discontinued some years ago because we have a new one, but unfortunately I don't have a picture of it. This is the heart of an EEG setup. It essentially serves, the purpose of it is to amplify the electrical brain activity that we record on the scalp, because that activity is so minute we need to amplify it dozens of times in order to actually be able to see something. And this amplifier is connected to a number of electrodes that are usually mounted in a cap that you can see in the middle picture, and all of these are placed on a participant's head. So this is a general setup. And on this slide, we see an output of an EEG recording over a duration of, let's say, 25 seconds in 5 different channels, 5 different electrodes placed at different positions, in different positions on the scalp. So this is essentially what we see on the screen when we do an EEG recording. This is also something you may have seen in case you've ever had clinical EEG performed on yourselves or somebody else. Right, so we've established now that EEG measures the brain's electrical activity, but what remains unknown as of yet is how is this of any use to us in language research. So what we normally do when we carry out EEG experiments in our linguistic lab is that we record brain's electrical activity while the brain is busy processing language. What does it mean? It means that we record the electrical activity of a participant while that person either reads something or listens to something. And by something, I mean language content. It can also potentially be while that person is producing language that is speaking, but that's something we'll talk a tiny bit about later. So in this setup, we have a participant who has an EEG cap mounted on their head with a number of electrodes. They are exposed to some language input, visually or orally, meaning through the ears, and the electrodes are picking up on their electrical brain activity and the changes therein at an extremely fast pace. So this can be at the level of milliseconds, and with the newest equipment it can be as fast, at least with the equipment that we have, we could get a new data point at 1/16 of a millisecond. This is so fast that we actually do not even record at this rate. We get a data point normally at the millisecond rate or even slower than that. So our goal applying EEG in language research is to observe how language processes unfold in real time. So EEG is for sure an online measure. And then the goal is also to characterize the processes that we observe. So yes, we can say, like in behavioral experiments, okay, there is a condition between A and B. But if you do a reaction times experiment, the most you can say is that condition A is different to condition B by being faster or slower. With EEG and with the application of event-related potentials, we can do actually more than that. We can really characterize or qualify a process by saying something like, okay, condition A is different than condition B, and it could be because there is more semantic processing in condition B than condition A. Now, the question is, how do we actually achieve that? How do we do that? Well, we do that by applying EEG in a certain form called event-related potentials. As I may have mentioned, the definition of EEG is that it is a continuous recording, meaning that if you go for a clinical EEG, your doctor is going to observe you and have your EEG recorded for maybe 20 minutes or 8 hours if you go for a sleep study. And this is going to result in one huge recording that is then going probably to be eyeballed. It really depends. With the application of event-related potentials, we sort of make the scope or the interest of the electric or the part of the electrical activity that we're interested in smaller. So what we do is that we try and take out only the bits of the electrical activity that are around the stimulus we are interested in. In order to make this more understandable, I'll give you a linguistics example from a semantic priming paradigm. I believe you all know what a priming paradigm is. In this case, we have visual presentation of a word on the screen. So we may see the word chair on the screen. After that, we see the word apple on the screen. And after that, we get a lexical decision task. We need to say if the word we saw last is a word or not. And let's imagine that it takes us around 600 milliseconds to say, yes, apple is a word. Of course, in such a paradigm, there are some examples where this target word would be a non-word. In a second condition, we may see the word pear first, and after that, following the word pear, we may see the word apple. Now, when we press the yes button, it may take us 500 milliseconds. Why? Well, because of the priming effect. Pear and apple are semantically related, And by activating the verb pair initially, we sort of pre-activate, just very crudely speaking, the whole semantic field of fruits, for example, in our brain. And this is why the access to the word apple is faster when we see it after the word pair rather than the word chair. Now, we can measure this with reaction times, but we can also do this with event-related potentials. How? We have a person do this experiment, their EEG is continuously recorded, but we send small pulses into the signal that are going to denote the visual onset, so the onset of the word apple on the screen. So in the continuous EEG recording, we are going to see markers that are going to tell us, okay, this is the onset of apple 1 and this is the onset of apple 2. What we are going to do next is we are going to cut out these chunks of EEG activity around the stimulus of interest, in this case the word apple. So we may cut out, let's say, 100 milliseconds before the word apple appear on the screen, and then another 1000 milliseconds or a second after the word apple appear on the screen. So we are going to do that both for apple. So what we are going to end up with then are two event-related potentials, one for apple 1 and one for apple 2. Mind you, then this is the electrical brain activity from, let's say, the visual onset of apple 1 and then another one for the visual onset of apple 2. How is this of any use to us? Well, we can try and overlay the two and check for any differences in activity between the ERP for apple 1 and apple 2. And if we overlay the two, we are looking for certain differences between them. What are these differences? Well, they are something known as components. For example, the N400, the P600, or the LAN, the MMN. I believe you've already encountered these abbreviations or acronyms, and this is something that we are going to talk about in a second. In order to better explain what a component is, I would like to start using an example from sentence processing studies, which means studies that use whole sentences as stimuli. So what does a typical sentence processing ERP study look like? Let's imagine a relatively simple study with two conditions. These two conditions would consist of sentences that are identical for both the neutral, that is the baseline condition, and the experimental condition. And the only difference in a neatly designed experiment would be in one place or in one word. What do I mean by this? Well, most often we use the so-called violation paradigm in sentence processing ERP experiments, meaning that the experimental sentence is either ungrammatical or semantically implausible. Most often this is achieved by manipulating a single word. So for example, we may have a baseline sentence such as Mary goes to school every day, and then we may have our experimental sentence containing a violation of grammaticality that would be something like Mary go to school every day. So there would be subject-verb disagreement. So that's what I mean by having one small manipulation in the experimental condition, usually at the level of a single word. Now, depending on what we want to do, we can choose to present this experiment as a listening experiment or as a reading experiment. If it's a reading experiment, what is most common is to present the stimuli sentences word by word on the screen, meaning that words will appear in isolation one by one in the center of the screen with a certain time between them. So, for example, I gave here SOA, which stands for Stimulus Onset Asynchrony of 600 milliseconds, meaning that the time between the onset of word 1 and word 2 in the experiment is 600 milliseconds. And this, of course, stands only for the visual modality. In a listening experiment, there is no need to do any sort of manipulation like that. It is enough just to have natural speech. Now, what we are going to do is we are going to time lock our signal to the critical word, and the duration of this event-related potential that we are going to create is probably going to be around 1000 milliseconds. A tiny bit before the onset of the word for maybe 200 or 100 milliseconds, and then another 1000 milliseconds from the onset of the word. After that, we are going to, well, there is a lot of pre-processing and there are a lot of methodological things to do there, but let us imagine that we are going to overlay these two ERPs, so one presenting a baseline and the other one presenting the experimental condition, and we are going to present a single graph with two waveforms or lines, and we are going to look for a difference between the two. How exactly is coming right up. The example that I am giving you here is from the seminal paper by Kutash and Hilliard from 1980. This is the first language-related EEG or ERP experiment ever carried out. The goal of the experiment was to look into how our brain reacts to unexpected stimuli in general, something that had already been done before in general neuroscience. The novelty of this experiment was that the authors decided to use language stimuli, meaning that they presented semantically unexpected sentence endings. So their baseline sentences were something like, and these are actually exact examples as presented in their paper. So a baseline sentence was something like, Mary spread the warm bread with butter, and their experimental sentence was, Mary spread the warm bread with socks. Now you're all going to agree that socks here is semantically implausible. It doesn't fit into the context. It's not predictable. It simply does not fit in. Now, if we cut out an ERP for butter and socks, and if we overlay them, if we overlay the two ERPs, mind you, I'm speaking mostly metaphorically. It's not as simple as this, but for the sake of explanation, this is how I'm doing it. We're going to end up with something like what we see in the graph below. We are going to see two lines. The full line is the baseline, and the dotted or the dashed line is the experimental condition, or represents the experimental condition. If you look at the two, we'll see that they are identical or completely cover or overlay each other up to some 300 milliseconds from the onset of the final word, which is the target or the region of interest. And then the two lines start to diverge, with the line representing the experimental condition being more negative. How do we know that? Well, there are two polarities, positive and negative, and in this case, negative is plotted up and positive down, which you can see on the graph given as plus and minus. So we see that the response, the brain's response to the semantically implausible stimulus is more negative, 300 to 600 milliseconds from the onset of the word, peaking at some 400 milliseconds. This regularity in the signal, which is something that we can observe in any or almost any semantically related experiment, is called a component. And the particular component that we see here is the N400. N for negative and 400 for simply peaking at 400 milliseconds. So this is what a component is. A component, and mind you, this is a very liberal definition, so I'm just doing it for the sake of making things clearer for you guys. A component is part of the EEG signal that has certain characteristics. It can be positive or negative. It has a certain distribution on the scalp. In the case of the N400, it's mostly central and parietal, but peaking in central regions. Most importantly, we can ascribe a certain function to the component. And in this case, broadly speaking, it's the processing of semantics. Now that we've established that there is a component that seems to represent semantic processing, we may start wondering, is there something like that for syntax? And let's say for now that there is, and that something is called the P600. So if you compare the two sentences given on the slide, the elected officials hope to succeed and the elected officials hopes to succeed, with the experimental sentence containing a subject-verb disagreement, we can see in the graph that the brain response represented by the dotted line is more positive than the baseline starting some 500 milliseconds from the onset of the word presentation. The duration is actually quite long. We can't see the end of it. But the most important part is that it starts at around 400 or 500 milliseconds after the onset and is positive. And this effect is a very well-known and established effect called the P600. You may be wondering why 600 now, because the N400 peaks or is the strongest at 400 milliseconds, but the 600 here doesn't make a lot of sense. It is for historical reasons. In one of the first experiments that reported the P600, the 600 millisecond was the time point. It's exactly the halfway of the duration of the effect. So it doesn't really mean anything. You just need to remember it as the P600. I told you on the previous slide that for now we can say that the P600 represents syntax. However, there's been a lot of development in the literature. We've started questioning whether this dichotomy between the N400 representing semantics and the P600 representing syntax is actually correct. And there's been a shift in the not even so recently, it's already been going on for 15-20 years, so there's been a shift away from calling especially the P600 a marker of syntactic or morphosyntactic processing. So there have been a number of studies that have to do with semantic processing that also elicited the P600 where the N400 may have been more expected, and due to this there has been some sort of refinement in terminology, and now we do not say that the P600 represents morphosyntactic processing anymore. We say that it reflects integration or repair and reanalysis processes. A repair process, simply speaking, is a process that turns ungrammatical input into grammatical one, and the idea is that the processes behind turning an ungrammatical input into grammatical input are those that result in the P600. And actually the exact classification or characterization of the P600 and to an extent the N400 is an ongoing debate in the field and is one of the most popular questions. So if you're planning to do research on ERPs in language, this is absolutely something you can do, even though it may be slightly more advanced because you really need to be familiar with the literature and have the historical perspective. Okay, so I hope that finally halfway through this presentation you understand what event-related potentials are, and now we're getting to actually the goal of this presentation, which is to tell you a tiny bit about how you should or you can design an ERP experiment so that you have a methodologically sound study. So let's start from the beginning, of course. Before running any experiment, you should have a developed research question or a hypothesis or a prediction or a name or a goal, whatever you would like to call it. So you need, before you start creating your stimuli and before you start conducting an experiment, you really need to have a research question in place. After that, there are several decisions that have to be made, and these decisions have to be made based on your research question and what it is that you actually want to achieve. So one of the first questions, in case you're doing comprehension, and by the way, I'll mostly be talking about comprehension because it is much more prevalent in the field. You need to decide about the modality of your experiment. Are you doing an audio or a visual experiment? There are reasons to do either. I prefer visual experiments because data are cleaner, but audio experiments are considered to be more ecological because in visual experiments we have single word presentation, which is not the way we read. So that is really entirely up to you. Both are present in the field. Visual experiments may offer cleaner data and they may be simpler to set up. But this really depends on you and your research question. The second thing to consider is, do we already know which components we are targeting? And at this stage, if you already have a developed research question, we should really know this. In case we're doing language production, we do not necessarily have to know it, but in case of language comprehension, absolutely yes. In the sentence processing field, we are mostly talking about the N400, the P600, and another component, the so-called ELAN, or the LAN, which I haven't really talked about. Now we have decided on our research question, we have decided on our predictions, that is the components we are targeting, we've decided sort of on the method. So for example, what modality we are going to use, what kind of analysis type. We also should decide on the participant group, but this is inherent to the research question. And then after that, we can go into more finer details of deciding the exact design of the study, the conditions that we are going to use, what these conditions are going to consist of, the stimuli that we are going to use, and what the format of our stimuli is going to be, and also the distribution of our stimuli, how we are going to present these stimuli to whom. That is, how many stimuli each participant is going to see, if each participant is going to see all our stimuli, and so on and so forth. Now, for those who are absolutely new to this and who may be wondering, okay, but what kind of research, language research, can I do with event-related potentials? Meaning, what kind of research question can I even ask? I have a couple of examples here on the slide. So most ERP research is related to neurolinguistics or psycholinguistics, meaning that we are interested in language processing in general. That is, we are interested in all cognitive processes going on in the brain while we process language. Now, what aspect of language? That can be anything. It can be morphosyntax, like I've already explained. So, for example, we may be interested in whether our brain processes different kinds of disagreement in the same or in a different way. So, for example, person disagreement and number disagreement, are they just classified as a sort of disagreement by our brain and the processed and repaired in the same way? We can look into semantics processing, pragmatics, phonology, any level of linguistic processing, essentially. And in addition to that, in addition to typical processing, we can also look into language processing in impaired or atypical populations. Then we can also apply ERPs in sociolinguistics research. For example, how we perceive foreign accent in our native language, depending on whether we know that somebody is a migrant or not, or a second or a heritage speaker or something like that. It's not really my field, so I'm coming up with examples on the fly. There is a lot of application of event-related potentials in bilingualism, multilingualism, for example, in control mechanisms. There is more research also into applied linguistics or second language acquisition, so we can track, for example, vocabulary acquisition in second language learners or the acquisition of a certain grammatical function in second language. We can also look into first language acquisition or development that can be typical or atypical. So essentially anything that can be experimentally achieved can also be done using event-related potentials. Regarding the populations we can study, there is not a lot of limitation. Um, EEG and ERPs are considered to be non-invasive techniques, meaning that they can be applied to vulnerable populations such as very young children, even newly born babies, elderly adults. So there are very, very few limitations. There are practical considerations though. So during the experiment, the participants should be as still as possible because any kind of movement may introduce artifacts and essentially prevent us from gaining any meaningful data from the signal. And in addition to that, depending on what exactly we are studying, our participants may need to be doing something consciously. And you can think of, for example, testing 5-year-olds on a 45-minute long experiment. That may not really work out, not because of EEG, but because they simply cannot focus for 45 minutes without a break, even with breaks, I would guess. Still, there are paradigms and components that are pre-attentional, meaning that our participants don't need to be paying overt attention to something. And this allowed us to carry out research with, as I mentioned, newly born babies and even with people in a coma. So if you're interested in this, you can just look it up. It's very easy to find. We can also test different neurological patients, so people with aphasia, dementia, multiple sclerosis, even Parkinson's disease, if their tremors are under control or if there are too many movement artifacts. What's very interesting is that we can look into groups with atypical or impaired language development, such as children or adults with DLD dyslexia, also with autism. We can also do longitudinal studies and look into what is going on during development. There are some components that I haven't mentioned before, but that can be looked into during the acquisition of literacy. We can follow what kind of changes happen in children's brains when they start to read and write, for example. And as I said, main limitations essentially are practical. If somebody cannot pay attention to an experiment, or if somebody has excessive bodily movements or jerks that they cannot control, then we cannot really do EEG. Related to one of the previous points, and that is that attention is one of the limiting factors, we have to think about the design of the experiment and conditions in terms of its duration and number of stimuli. So you may be thinking, okay, but if somebody cannot do an experiment that is 45 minutes long, why would we even create such an experiment? Why not create a 10 or 15-minute experiment? Why does somebody have to see 240 sentences and not 24 sentences? Well, like in any psycholinguistic study, there is a certain demand that is either related to statistical power or something additional to that. And in this case, what we are limited by is the nature of EEG data that we obtain. EEG data are quite noisy, meaning that they contain information that is not related to what we are interested in. So in addition to containing brain's electrical activity that is a response to our experiment, there is also electrical activity that has nothing to do with it and is coming from external sources or internal sources that we're not interested in. So due to the noise or noisy data, we need to have as many stimuli as possible. And then finally getting to the point, which is, okay, but how many stimuli? And this is something that I kept being asked by my students and people I collaborate with. The answer to this is really, it depends on the component you're interested in. As a rule of thumb, if you're doing a sentence processing experiment with the N400 and the P600 and the LAN, I would say that it is enough to have 20 stimuli per condition. Now, 20 is not a magical number. If you can have more than that, that's fine, even though I've recently seen a paper that claims that having more than 20 stimuli does not actually contribute anything except extra time for the experiment. And if you look at previously published papers, you would see that most of those report around 20 stimuli. And what I mean by 20 stimuli, I mean that if you have an experiment in which you have grammatical and ungrammatical sentences, let's say only two conditions, each participant has to see 20 grammatical and 20 ungrammatical sentences. I'll come back to this. If you're interested in working on other components such as the MMN, please have a look at the previous studies, several or as many as you can, and try and see what the average number of trials is. This is not always simple because people sometimes report lists, sometimes they don't. I will talk about this in a second. So sometimes it may be difficult to actually elucidate the number of experimental items that they used. When it comes to the MMN, just to illustrate the difference, unlike P600 and F400 studies, MMN study may work with hundreds, if not thousands, of stimuli. Let us have a look at a sentence processing experiment that looks into subject-verb disagreement. Let us imagine that this is a simple experiment with only two conditions, a grammatical and ungrammatical condition, such as in 1 and 2. Now, I mentioned on the previous slide that each participant should see at least 20 items of each condition. For this particular experiment, it means that each participant should see 20 grammatical and 20 ungrammatical sentences. If you look at these sentences, we'll notice that they are identical, except for this small manipulation that causes violation. And this is a good design. This is how it's supposed to be. However, there is something like, let us say, an unspoken rule that one participant should not be exposed to both versions of the same sentence, meaning that we shouldn't have one participant read both the grammatical and the ungrammatical version of the sentence "Peter goes to school every day." This slightly complicates things, meaning that we actually have to create more stimuli than one participant is going to see. What does that mean? Well, in this case, if you need a participant to see 20 violated or ungrammatical and 20 grammatical sentences, we need to create experimental sentences in total. That would be 40 pairs of grammatical and ungrammatical sentences. And why is that? Well, if we only had 20 sentence pairs with 20 grammatical and 20 ungrammatical sentences, one participant would either see all sentences, so all sentence pairs, or we would have a participant see 10 grammatical and 10 ungrammatical sentences. And none of this is desirable. If they only see 10 sentences in each condition, this is probably not going to be enough for us to actually see a component. And if they see all versions, both grammatical and ungrammatical versions of all sentences, methodologically, this is not quite acceptable. This is one of those rules that people sort of ask you to stick by, even though there isn't a lot of experimental evidence that indicates that it may be doing something. So let us consider that this is sort of good practice in the field. The idea is that we can create some sort of expectation strategy, expectation-based strategy. If we know that we are going to see each sentence once in a grammatical form and once in an ungrammatical form, we can learn to expect which sentence is going to show up. Let us say that there is a potential explanation as to why methodologically we shouldn't have all participants see all versions of our stimuli in case they're built in such a way. In order to prevent that, we need to create something called lists. And this means that each participant is only going to see one list. If we have two versions of one sentence, that means that we need to create two lists. If we had four versions of a sentence, we would need to create four lists. Here is an example of what exactly it would look like. So in case we create 40 sentence pairs, 40 grammatical and 40 ungrammatical sentences, the distribution of items would be as follows. So in list 1, for example, we would take the first 20 grammatical items, and for ungrammatical items, we would take the other 20 ungrammatical items. And for list 2, we would do it the other way around. This sort of item distribution is called the Latin square, the Latin square design. Another good practice is that in case we have multiple lists, we should have and try the same number of participants per list. This is not mandatory, it is only a good practice, but yes, if possible, we should try and have the number of participants balanced across our lists. Also, the lists have to be identical in terms of the number of items, conditions, and essentially everything. So the lists need to be essentially identical, have an identical distribution of items. So when it comes to what our stimulus should look like, I am going to give you a couple of very general tips. The first tip is to control, control, control. You may want to control for frequency, for sentence length, for animacy. It really depends on your research question. It is not absolutely always necessary to control for all possible and impossible psycholinguistic variables, because that is for sure impossible. So in an experiment such as the one I presented on the last slide, where you would have something like John goes to school every day versus John goes to school every day, there is no need to control for frequency because all the words that we use are the same. On the other hand, if we were to talk about an N400 experiment, such as the original 1980 experiment, we would really have to take frequency into account. So if we have something like Mary spread the warm bread with butter, or Mary spread the warm bread with socks, we really want to make sure that butter and socks are of comparable frequency. We don't want to use a word in the experimental condition that is of much lower frequency than the word in the baseline condition, because that can contribute to the semantic demand that is necessary in order to process the sentence. So some of these variables that we need to control, or most of them, are actually determined by our research question and the design that we are employing. What I also like to recommend is to have a balanced design. So there are some things that we don't have to control for. So for example, sentence length is something that it's extremely difficult to achieve, to have all your sentences of the same length, and it's not really necessary. But if they vary in length, it may be a good idea that they vary with some sort of consistency. So for example, to have an equal number of sentences with this many words and that many words, so as not to end up with having 5 sentences that are 6 words long and 25 that are 15 words long. Also, in case we are talking about sentences, what is good to have is to have sentences or items that are structurally as similar as possible. So consistency is also another point that we have to keep in mind. So control, balance, and consistency. Lastly, you should probably avoid, and this holds for any psycholinguistic experiment, not only for an EEG experiment, you should try and avoid putting your target word at the end of the sentence. Now, this is sort of a contentious issue. Some people claim that there are additional effects happening at the end of a sentence called wrap-up effects that can actually interfere with the desired experimental effect. So this last tip is essentially just a precautionary measure. Don't do it unless it's absolutely necessary. If you're interested more in wrap-up effects, you can read articles by Harcourt or by Stowe. Here is a small exercise. I'm going to give you some time to do it. Since this is a recording, I think 10 seconds is enough. So the solution is like this. There should be 3 lists because these 3 sentences are more or less identical, except for 1 word or 2 words in version 3. Each participant has to see 20 sentences in each one of these conditions. So each participant is going to see 60 sentences, but that means that we have to create 60 sentence triplets, which in total is 180 sentences. One of the questions that keeps being raised constantly is the question of fillers. Do I have to have fillers in my experiment? Fillers are additional items besides experimental items that are present in almost any experiment. So the norm in the field or the custom is to always have filler items. Their goal is either to distract the attention of the participant from the research question or to try and prevent the participant from creating some sort of guessing strategy. It really depends on the experiment, but the bottom line is the same. Yes, each experiment should have fillers. They should be something like 1/3 or 1/2 of all items. That somewhat depends on your design. If you already have a lot of experimental items, it is all right to have a fewer number of filler items in that case. What we should try and have in our experiment is the principle of economy. That is, our experiment should be as short as possible because that is going to guarantee data of high quality. People's attention decreases over time, as we know, and actually that is something that we can see in the quality of the EEG signal. So let us say that student without any language or reading problems should be able to do a sentence processing experiment, so read sentences for something like up to 30 minutes. This is approximately 240 sentences, which should be enough for a relatively large experiment with 4 conditions. Everything after that is already going to cause some strain. It's not impossible, but it is absolutely not desirable. One way of compensating for the length of experiment or alleviating for the fatigue effect is to organize your items into blocks. Blocks can mean different things in different fields, but in this case, it means just organize sentences in groups. So for example, if you have 240 sentences, we can organize them in groups of 60, have 4 blocks, and then after each block, we can tell our participants, okay, take a 2 or 3 or 5 minute break just to rest from the experiment and think about something else. So this slide actually gives an example of how experimental items can be distributed. One more thing to keep in mind is that when you distribute your items, you have to be careful that, so there needs to be a balance. You have to have the same number of items of the same kind in each block. So you shouldn't have more items of condition 1 in block 1 than in block 2. So there should be an identical number of items across blocks. So it should be as balanced as possible. When it comes to the presentation of items, a lot of presentation software actually offers to sort of randomize items for you. What I recommend is that it might be best if you pseudo randomize items yourself, meaning that you create or you put items in such an order that it's impossible to create a guessing strategy. Now, a couple of points that are rarely raised, but that actually may play an important role, is when you introduce your participants to the experiment, do you want to reveal your research question or not? Again, this depends on your research question and the component you're targeting, but for most P600 and N400 experiments, this is actually not desirable. So what we are trying to do is we are trying to actually obscure the research question, and we are trying to prevent our participant from guessing what the research question is. One of the reasons for that is probably that we do not really want our participants to create any sort of guessing strategy, or if they figure out what the experiment is about, they can just um, go through it without thinking too much about it. Now, if we are talking about a comprehension experiment such as listening or reading, how can we actually make sure that our participants are indeed paying attention? Um, we usually achieve that by introducing a behavioral task in addition to the EEG recording. In case of sentence processing with ungrammatical sentences, this can be a grammaticality judgment task, or it can be a content question. And mind you, it's been shown that the nature of this task can actually affect the results. So it's been shown that uh, the grammaticality judgment task can actually enhance the P600 component, probably by making people focus more on the grammaticality aspect of the task. So if you are designing an experiment, you should definitely look into this and check out if there is previous literature on what kind of a behavioral task is most suited for your kind of paradigm and research question. In addition to all the methodological aspects, there are certain technical aspects that we need to think of, and I'm just going to almost gloss over them. Um, I'm not going to go into detail. I'm just going to say that you should think about how many electrodes you want to use, because this is going to affect um, how long the preparation is going to take, but also the amount of data you're going to end up. For most studies nowadays, if you're not doing any kind of fancy analysis, if you're just focusing on the N400 and P600, 32 channels are the standard, as well as using the average of the mastoids for the reference. So this is the standard in the field now. Now, this is a small exercise. In case you are tech savvy and you understand what the reference electrode does, which is not something I'm going to go in detail in case you don't, think about why the average of the mastoids is the gold standard nowadays, and why, for example, using a CZ electrode may not be the best choice. And my final advice, when you've finally designed your experiment and you programmed it, and now you're going to start recording it, my advice is that each trial counts. Try to minimize, prevent any data loss while recording. Observe your participants and the recording all the time. Don't leave the room while the recording is ongoing. Why am I saying this? Well, you may see some things that can actually be repaired even while recording is in progress. You may see that an electrode is flatlining, and then you may be able to fix it, maybe even during the recording or during a break. Or you may notice that your participant has started humming and you want to alert them and tell them not to do that. There are other applications of EEG and ERPs. There is one more application of event-related potentials that I didn't talk a lot about, which is the application of event-related potentials in speech production, language production. This is maybe not as mainstream as comprehension because it is technically more challenging. As you know, speech production requires the use of muscles, which can introduce a lot of artifacts in the EEG recording. There are ways to circumvent this. So, for example, we can record only up to the point of uh, the onset of speech production as to avoid these artifacts. But in order to be able to do that, we need to do quite a few things, which makes the whole process much more technically and technologically challenging. Now, there is another application of EEG. There are a few other applications of EEG, but another one that is very well known is the so-called time frequency analysis. ERPs are the so-called time domain application of EEG, but we can also look into the time frequency domain, which, unlike EEG, looks into different frequency bands, such as the alpha theta band, and their changes instead of looking at the changes of voltage that represents the electrical brain activity. So the time frequency analysis is something that is slightly more demanding than the traditional ERP analysis, but it is widely done, and there are many, many studies that apply this kind of analysis to EEG data. The nice thing is that most of the time, the time frequency analysis is compatible with the ERP analysis, so both can be done in the one go. However, as you've already seen, it's almost a full hour, and they only managed to talk about event-related potentials, and there are so many more things that I would like to share with you. So introducing the time frequency analysis would have been too much. So maybe some other time. And finally, to wrap up this workshop, EEG in the form of event-related potentials has been used in language research since 1980, the seminal article by Kutesh and Hilliard. It is relatively demanding. It requires a complex setup in which we have many methodological choices, and all of these choices have some desirable or not so desirable consequences. So why did I focus today on the methodological choices rather than something else? Well, if you think about it, data pre-processing, and statistical analysis, all these steps can be done as many times as we like. We can just tinker with it. We can change one parameter, and if it doesn't really work out, we can just repeat the whole process. But if you make a methodological error, such as an imbalanced paradigm or too few items per condition, we can almost never fix this. So we cannot really fix our data after we have done data collection. Something that you will find a lot in the ERP literature is that there is a general rule for data acquisition, which is to try and prevent noise instead of removing noise in the pre-processing stage. This can also be applied to methodological choices. We need to make right choices before collecting data rather than try and fix those post hoc, because as I told you, most of the time it is actually impossible to fix problems caused by faulty design. This is the end of the workshop. Thank you very much for your attention. In case you want to get in touch with me, my email address is on the screen. Thank you again.